So for 18.4, we're looking at bacteria and archaea. They're similar. Bacteria and archaea are single-celled organisms. They're both prokaryotes, but they do have differences too. So even though you can consider them both bacteria, they do have different kinds of DNA and different chemicals around their cell walls, and so they are quite different from each other. It's kind of like saying, that a beetle and a dog are both animals. And that's true. You can consider a beetle an animal just like a dog, but they also are they're very different from each other too. So it's similar when we say bacteria and archaea. They're both kinds of bacteria, just like an animal is both a beetle and a dog, but they are quite different as well. So archaea are bacteria that live in harsh environments. Has anybody been here before? I recognize this place. This is Yellowstone. Now, this is a hot spring in Yellowstone. And it's really amazing the colors you see there. These rainbow colors are from bacteria that live in the hot water. So it's near boiling, it's 200 degrees, but the bacteria can still survive that temperature. So archaea are bacteria that can survive very hot temperatures, 200 degrees or so, and incredibly cold temperatures as well. So you can find them in the Arctic at 5 below zero. You can find them in hot springs, find them in the mountaintop with very little oxygen. You can find them in salt water where everything else dies. So they are able to survive in almost any kind of environment here on Earth. They've even been found in nuclear reactors. So even radiation kills most everything, but they've even been found there. So these are prokaryotes. Bacteria are prokaryotes, and they're very widespread on Earth. There's more than 1 billion kinds of bacteria that we've identified, and probably a lot more that we don't even know about. And these are some of the shapes of bacteria and the arrangements they might take. So you might see some as a spherical shape. This is what causes staph infection. Staphylococcus, we just call it staph for short. Streptococcus causes strep throat. Pneumonia is caused by Streptococcus pneumoniae. Escherichia coli, or E. coli for short. Helicobacter pylori causes ulcers in your stomach. You don't need to know all these down here. It's just for your information at this point. So don't worry about copying down these shapes. I will tell you on the next slide what to copy down, but it's just for you to understand that they're very diverse shapes, different arrangements of bacteria, but these aren't ones you have to know. So on this slide, just know these top 2 bullet points. So there's 1 billion kinds of bacteria, but there's 5 with 30 zero cells of bacteria on Earth. So bacteria far outnumber any other type of cells, plant, animal cells. If you were to get a scale and weigh all the bacteria and all the animals and plants and humans, the bacteria would far outweigh the humans, both in mass and in number. There's only about 7 billion people on the earth, but there's a billion just different kinds of bacteria. There's different ways we can group prokaryotes. And we talked before about archaea, how they're bacteria that can survive harsh environments. We can also group them in this way. There are some that need oxygen to survive. And others that are poisoned by oxygen. So the ones that are poisoned by oxygen are called obligate anaerobes. Obligate means, obligatory. You have to do something. You're obliged to do something. So obligate anaerobes are ones that cannot be around oxygen or they'll die. So you'll find these in swamps, the bottom of swamps. So down at the bottom, there's no oxygen, they're in the mud, but you still need to have bacteria down there to decompose dead animals, dead plants, unlock the nutrients that are in those areas. And so they're down at the very bottom of the swamps and marshes. And as they decompose other things, they unlock gas and nutrients. So you'll find these bottom of lakes and ponds and marshes deep in the ground where there's no air. They're also in animals and humans' digestive systems. And in a human and animals, they make vitamins. For us, they make vitamin K in our intestine, which is important for blood clotting. If they're in the soil, they unlock the nutrients in the soil and bring them back to the plants to be used again. And the other kind, actually, this should be aerobes, not anaerobes, sorry. So obligate aerobes need oxygen. Anaerobes don't need oxygen. Aerobes need oxygen. So these would include bacteria that live in the lungs. Most of these are, many of them are harmful to humans and animals. So some bacteria that cause tuberculosis and leprosy are aerobes. So these are the ones that can live in the lungs and cause tuberculosis. Leprosy is a skin disease. It can also attack the nerves. And so over time with leprosy, the nerves are attacked and a person becomes numb and they don't know that they're being damaged. And so with leprosy, the biggest concern with leprosy is that the nerves have been deadened. And so a person doesn't know they're being hurt. They could be walking on their foot the wrong way and maybe breaking their bones or scratching themselves or causing a cut. And they have no way of knowing that because they don't feel the pain there anymore, and that will cause more damage than leprosy usually.
Miss Polly.
Yes.
But like, In didn't aren't their skin like rotting away for leprosy?
Exactly, and so that's true. Definitely, you're going to see their, especially their fingers and their toes. They do rot over time. That's true. But
And
usually
is
that's not
that
what kills them. Usually, what kills them is their lack of pain, and then they either bleed to death or they cause a severe injury they don't know about. But definitely, the rotting away is also a concern, a big concern in leprosy.
so it can like damage your cells to where it rots away and has no like sense of pain?
Exactly. Yeah, it attacks the nerves too. So it goes from the skin to the nerves. And it's a fairly slow process. And so you can live a long time with it. Generally, what kills the person is not the rotting itself, it's the lack of pain. And they then cause a severe injury that they don't know about and they can't treat it in time. Okay, the third class is facultative anaerobes. And so to have a faculty means, or facultative, sorry, facultative anaerobes. So to have a faculty means you have the ability to do something. So these are ones that facultative aerobes can survive with or without oxygen. So they have the ability to either be in an oxygen environment or a deoxygenated environment. They can live either below the ground without oxygen or they can live above the ground with oxygen. So obligate means they have to. So obligate aerobes means they have to live with oxygen. Facultative aerobes can survive with or without it. So I mentioned before that bacteria and archaea are similar to each other in structure. They look very similar. They are single-celled organisms. They're both prokaryotes, but they do have different chemicals that make up those cells. So we'll talk more about that in a little bit. But they do come, bacteria come in three major shapes. Archaea can come in more diverse shapes. We'll talk about archaea in a little bit, but these are the three basic kinds of bacterial shapes that you're going to see. So there's rod shape, it's in the middle on this picture right here. Bacilli is the plural, bacillus is singular. So bacillus shape or rod shape. Second kind, which you see on the right, is spiral. It also could be called spirilla or spirochetes. And the last one, which is on the left-hand side, is spherical. Cocci is plural, coccus is singular. And of these three basic shapes, they can be different arrangements. You might see some line up. Others can be in more like a cluster. Others live on their own. So some live in colonies, others live by themselves, and that will determine the properties as well. So these are, again, these are bacterial shapes, archaea. I'll show you the shapes later on, but they're more complex shapes than these three. And that's just another picture of the same thing. So this just shows you another viewpoint of the rod shape, spherical shape, and spiral shape bacteria. Those are the bacterial shapes. These are archaea. And you can see in here that, they do have some of the same characteristics. You do have some that are spherical, some that are rod, but also we have other complex shapes as well. Some with flagella, these tails at the end, some with several flagella, some with various shapes, like maracas or, more like cylinders. Again, you don't need to know this as far as, all these shapes. Just know that archaea does have many different shapes, more than bacteria does. So you can see right here, this one causes salmonella. This one causes pertussis or whooping cough, diphtheria. This one causes food poisoning, botulism, tetanus, pneumonia. We already showed that one. And then stomach ulcers, staph infection. So let's go through a few. Now this one actually should not be right there, so that's strange. Well, so I'll have to go with that. Let me find this really fast if I can. Gonna fix this. I lost a slide for some reason. Let me go ahead and fix that. If you want, you can write down these first bullet point right here. I'll just add this picture while you're doing that. Okay, here we go. All right, so similar structures in both bacteria and archaea. One structure they both have is a plasmid. And a plasmid is a small piece of DNA that can replicate separately from the main DNA. So a plasmid looks like usually a circle, and that's what you can see. It's in blue on this picture right here. So the bacteria has the DNA, in this case it's red, it's all bunched together. There's no nucleus in prokaryotes, so it's just all throughout the cell. But a plasmid is a small segment of DNA that's broken off from the main DNA. And it can be copied separately from the main DNA. And that's what you see in this picture. So a plasmid usually is for one gene. It could be for immunity from antibiotic, for example. So this one gene right here could make this bacteria immune to certain medications. And we'll talk about more about why plasmids are important later, but for now, just know that a plasmid is a small part of DNA that's broken off from the main DNA. A lot of bacteria have flagellum. So flagellum is singular, flagella is plural. It's a long whip-like structure for movement. And you might see one, you might see several on one end, you can see some on both ends, or all around. Generally, the more that they have, the faster they're going to go through the water. And they're meant for swimming through water. They're kind of like a motorboat, the propeller on a motorboat. It spins in a circle as they swim through the water. And the last structure that bacteria and archaea have in common is a pilus, that's singular, or pili, plural. These are small hair-like structures that let bacteria attach to a surface and other cells. So these are kind of like the arms on the bacteria. They can pull things to themselves, pull food, pull other bacteria. or just attach to a surface and hold on to that surface. This is one reason why plaque on your teeth is so hard to get off. Plaque is built up of bacteria, and when it sticks to your teeth, it has this sticky outer coat. So this yellow right here can be made of sugar on bacteria and very sticky. And so that sugar capsule sticks to the teeth, and then these little arm-like structures can also pull onto that surface. And plaque is very hard to get off because it's just so sticky and stubborn. And that's why flossing is important, because flossing is the best way to remove plaque from the surface. Brushing won't do it. Bacteria are just too sticky. Just another picture of the same thing. You can see the pili on the outside, flagellum right here. On the inside, this is DNA, the purple. And so mostly all you see in a bacterial cell is DNA, along with some ribosomes, the blue dots right here, and then a plasmid, which you can see the circle right here. Don't worry about writing this down. You don't need to know this. The book goes over it, but it's not really worth going through in detail. So just write down this top bullet point and we'll just move on from there. Just know that bacteria and archaea have molecular differences. And so they have different chemicals in their cell walls. They talk about peptidoglycan, which is a chemical you can see in yellow on here. And the amount is going to differ between archaea and bacteria. We won't really go through that in detail. Just know this top bullet point that they do have different chemicals that will give them different properties. So one property that they have is they stain differently. If you add a stain to archaea versus bacteria, you're going to see that there'll be different colors. And so some will stain a red color, some will stain a purple color. And that will let scientists know what kind of bacteria they're dealing with. If you have an infection, they can know what medicine to give you. Because archaea will not be affected by the same bacteria, but the same medicine the bacteria is. So if it stains red, a certain medication will kill that one that won't kill the purple one, and vice versa. You don't need to write down any of this. Just know, the idea behind it, that the different chemicals on archaea versus bacteria let in different chemicals into that bacteria. And that means it can be immune to certain medicines or not immune to them. And by staining these ones, you can know which ones are immune and are not immune to a certain medicine. Now, both bacteria and archaea are going to divide in half to reproduce. It's called binary fission. So we talked about this before, that one cell right here will duplicate the DNA and then divide in half and separate to make two identical cells. So at the end, each daughter cell gets an exact copy of DNA. Each cell is identical to each other. They're basically clones of each other with same exact traits and DNA. So if that's all that happened, every bacteria cell would be the same, because there would be no variation in bacteria. They'd all would reproduce with twins, and you would have no variation in the bacterial community. But a few things happen to make there be variation. One thing is mutations. They're very prone to mutations. And so even though they are supposed to copy exactly, lots of times there's mistakes in the copying, which will make slight differences between the two cells. Besides mutation, conjugation also will result in different traits being added to other bacteria. And so these cells can join together, making a bridge, and they can pass traits through these bridges. So it's really amazing that a bacteria cell can somehow sense that one bacteria doesn't have a trait that they have. And it can sense that and give that trait to the cell that doesn't have that trait. And we have no idea how they know this. Bacteria don't have brains, they don't have eyes, they can't sense things like animals can. But somehow they have the ability to know when a certain bacterial cell needs a trait, and it will give it to them through conjugation. So this shows the process in more detail. So this bacterial cell on the left-hand side has a plasmid. It has a trait that this one doesn't have. So it might be immune to antibiotics. And it can sense that this one needs that trait. And so it will build this bridge using a pilus. It will build a bridge between the two cells, pull them together, copy the plasmid, and give it to the other cell. So at the end, they both have that same trait. And it could be a trait for being fast. It could be a trait for having a tougher cell wall, for immunity to antibiotics. But it's always a beneficial trait that they pass on to other bacterial cells. So that's one way bacteria can survive, by adding traits in the colony. They can also survive by forming endospores. It's a very thick, specialized cell wall. It's designed to survive harsh conditions with no food, no water, very cold or very hot conditions. You don't need to know the process down here. It's just to show you what it looks like at the end. Just know that endospores are just cells that are for hibernation. They have a very thick cell wall, and they're meant to be dormant and inactive during harsh conditions. So the end result is the DNA with a capsule that's very sturdy and able to keep in water and live for a very long time in bad conditions. Some endospores can make animals and humans sick. One example would be endospores in the soil. Anthrax, which are these little blue capsules, can live as spores in dry soil without any nutrients. And then it can go onto a plant, which animals could eat. And then when they eat it, they get the disease in their body, it becomes active, and the animal dies. And then they pass the bacteria back into the soil. As they die, the bacteria becomes inactive again as spores until they meet a living being, which then become active again. So it's a cycle between being endospores and being active in an animal or human. So anthrax is a very rare disease. It's not very common at all, but it does happen sometimes for animals or plants in certain conditions. In spore form, it looks like this. It looks like a white powder. So anthrax spores, these would be the dormant form of anthrax. Now, if you were to ingest this, then it would become active again and it would start to divide. But in this state, it's dehydrated and it stays inactive, doesn't reproduce. It just stays in place until it has the right conditions to survive and start to grow. Okay, so that's it for today for 18.4. Are there any questions before we go on? Okay, homework is on page 524. It's 1 through 6. And this will be our last lesson for this week. Then next week, we're going to go back on Tuesday to 18.6 and skip 18.5. I think I mentioned this before, but if you want credit for your homework, you can submit it to me and I will give you credit for your homework. I'll start to add homework grades starting next week. But from here on out, if you want to scan a copy of your homework and give it to me, I can give you credit for that.
If we did it on a Google Doc and we send that to you, would that also work?
Yeah, Google Docs is fine.
Okay.
Miss Holly, how would you do number 2?
Let's see, for number 2, why are bacteria and archaea classified in different domains? So a domain is just a different group. So why would they be in different groups and not in the same group? That's what they're asking. So we talked in the notes today about how they're different. How are bacteria and archaea different from each other?
What is it asking for number 3 about the genetic engineering?
Genetic engineering means adding genes, changing genes. One example would be taking bacteria and adding the gene that makes insulin and using that bacteria to make human insulin. So we can take the human gene that makes insulin, give it to the bacteria, and it can start making insulin for us so we can use in medicine. That's one example of genetic engineering. So they're saying prokaryotes will take up foreign DNA. And so they'll do the same thing that we do for them. They will take up different genes on their own and get new traits. And a scientist can do the same thing and give it new traits as well.